Hello, and thanks for joining us today for the latest The Scientist webinar. My name is Charlene Lancaster, Associate Science Editor, and I'm here to moderate today's event. I'm very pleased to introduce our speaker, Claire Gustafson. Claire Gustafson is an Assistant Investigator in Experimental Immunology at the Allen Institute in Seattle, Washington. Her research focuses on understanding the mechanistic underpinnings of immune aging in humans, utilizing high-dimensional techniques in conjunction with the development of novel model systems to accelerate translational human immunology research. A very warm welcome to you, Claire. Following the presentation, we'll have a Q&A session, and we welcome any questions that you may have. Please submit your questions using the Q&A system to the side of the live stream player. So without further ado, I will now hand over to Claire to begin her talk.
Hi, my name is Claire Gustafson, and I am an assistant investigator at the Allen Institute for Immunology based in Seattle, Washington. I am really excited to be here today to be able to tell you about some of our recent work focused on using multiomic profiling of human immune cells to actually be able to chart the human immune system and how it responds across age. By 2100, the global population is estimated to exceed 2.4 billion people over the age of 65 years old. So why is this so important to think about that the global population of older individuals is about going to double from today? This is actually really critical to think about when we look at disease susceptibility across age. And in particular, when we look at these more advanced ages or older ages, which is usually delineated about at 65 years of age, we see that there's significantly increased disease susceptibility for a variety of different diseases in older individuals. This includes viral infections, cancers, development of autoimmunity, and neurological diseases. And so it's very important to think about how we can mitigate this susceptibility to disease to bring down this burden as we have a globally expanding aged population. In particular, what we also know in this older age group, we actually have a longitudinally decreased protection from vaccination, one of our key methodologies for keeping people healthy. And so why does this happen? Why do we have increased disease susceptibility, reduction in protection against vaccines in older individuals? And so this is where the immune system comes in. And so what we know from many, many years of work focused on looking at the immune system is that its function actually changes across the course of one's life. So early on, the immune system is actually in development. It takes time for this immune system to fully develop. And then there's a period during adulthood where its function is maintained. And it's maintained long-term over the course of decades. But then there becomes a point where actually this maintenance breaks and the immune system and its function starts to decline. And this is actually, this decline is specifically contributed to our increased susceptibility to disease as we get older. And so really at the start of this work, we wanted to understand what are the age-related changes in immunity that are specifically happening that might link to dysregulation in immune function and increased disease susceptibility. So at a high level, we're really trying to understand if we can find mechanisms or ways that we can actually target eventually in the clinic that might prevent increased disease susceptibility in older age. All right, so once we think about this in that context, how do we do this? How do we actually do a study where we can ask these questions and gain insight into a time where we don't know when we're going to start to decline? And so I'll tell you how we approached this. So really we started this collaboration, this amazing collaboration with a group also located here in Seattle, Washington at the Benaroya Research Institute. Our colleagues Jane Buckner and Kate Speak spearheaded this effort in a consortium called the Sound Life Project. And so this was a collaboration to recruit individuals into our studies focused on looking at the immune system and its response in healthy people over the course of time, but actually at this point of immune transition. So in people prior to what we will classically consider old age. And so here we recruited two groups of people. We recruited younger adults who were 25 to 35 years old, as well as older adults in the 55 to 65 year category, right before where we would call them actual older adults, which is 65 and above. These individuals were enrolled in the study, and we enrolled almost 100 people, so it ended up being 96 in total. And they came in over the course of a 2-year time span for multiple different blood draws in where they actually also received the flu vaccine. And this is actually really critical because this vaccination is a way for us to understand how the immune system functions in response to something like a vaccine and or, if you think about it, an infection. And so really we're using the influenza vaccination as a tool to monitor these people's immune function. So this was an amazing endeavor. You know, people came in, this started in the middle of COVID, and we were actually able to continue it to enroll this large number of people. We can study now longitudinally at maintenance periods and as well as with the flu vaccine. In addition to this, at every time point, we did multiomic profiling in which we took the cells from the blood of these people. We did whole blood RNA sequencing, single cell RNA sequencing, spectral flow cytometry, mass cytometry. We also collected their plasma, did O-link proteomics, and CMV and flu serology. Really doing a comprehensive multi-omic profiling of these individuals to characterize their immune cell types, states, and responses over the course of time and across age. So utilizing, in particular, our single-cell RNA sequencing data sets, one of the first steps we took was actually to go back an entire step. And what we did is we ended up pooling single-cell RNA sequencing data from 108 donors from our collective cohorts that included individuals, healthy individuals, that were 11 to 65 years of age. This ended up amounting to a 1.8 million peripheral blood mononuclear cell data set. At the time, this was one of the largest data sets on human immune cells to date. And what we ended up doing was finally defining the immune cell subset present in healthy human blood. This is really critical because this allows us to then apply this reference across the board to all of our other data sets, whether it be in the context of health or disease. In particular, what I'd like to highlight of this data set is we ended up going down to 71 distinct cell types at our most finest resolution. That includes a variety of different types of T cells, B cells, NK, monocyte, and DC populations. We can then actually use this information to define really clear subsite gene markers. We can look at the frequencies of these cell subsets, and I'll show you actually in more detail how we ended up diving into this to answer some of our immunological questions. I'd also like to highlight that we've built out some data visualization and exploration tools specifically for this atlas. And so if you scan the QR code down below, it'll take you to the links to let you actually explore more of what went into building this immune health atlas. From the building of this atlas, what we did is we went on to utilize this atlas and do cell label transfer onto our entire Sound Life cohort. This amounted to more than 800 individual samples from our 96 donors across the course of time. And after QC and all of our quality control metrics and consolidations and doublet removals, ended up being more than 13.7 million PBMCs from this data set. We simultaneously ran multiomic profiling on a secondary data set that, instead of looking at this transitional point, actually spanned across it. And this is our immune aging cohort. And this ended up being from 234 healthy adult donors from single time points, cumulative ranging from the age of 40 to 90 years of age, more than 3.2 million PBMCs. And from this data set, collectively, we were able to start asking our questions about immune aging. In particular, understanding how do immune cell types and/or states change with age. So one of the very first analyses we did was actually to confirm a numerous amount of literature that demonstrates the loss of naive CD8 T cells as a hallmark of aging. And this is just highlighted here on the left, in which we looked at frequencies of the different T cell subsets, whether it's the CD8 compartment in green above or the CD4 T cell compartment below. And here what you can see is that the core naive CD8 T cells defined in our atlas show a remarkable decline with age. Now, this is actually very interesting because we don't see this similar phenomenon happening in the CD4 compartment, and I think this is much less appreciated across the course of aging, where the CD8 T cell compartment and the naive cells in particular are much more susceptible to loss over the course of time. And so our next question from this, great, we can confirm previous literature, but how do actually cell states change across age? And this is what we were really particularly interested in. And so now, using our Immune Health Atlas, we were able to apply this to our very large Sound Life data set and actually looked how the RNA profiles of 71 different cell types in the peripheral blood changed across age in our cohorts. These findings are summarized here in this bar plot, in which what we're looking at is the different RNA changes that happen with cells when you look at young adults versus our older adult cohorts. And in particular, you can see a lot of green and blue bars. These are colors for different T cell subsets. And what we can find is actually there is a significant amount of transcriptional changes happening in the T cell compartment, in particular focused on naive and early memory T cells. One of our next questions was actually focused on if this transcriptional change we're seeing in the T cells is actually stable over time. So is it a stably maintained transcriptional reprogramming? And in order to do this, we utilized the longitudinal nature of our study to look at transcriptional programming in T cells across the course of up to 600 days within individuals. And as you can see here on the left, we're highlighting one example in which older adults are in gold and younger adults are in teal, where we quantified this reprogramming in terms of an RNA age metric. You can think about this as a molecular clock for aging, in which it's a summary metric for all of the age-related changes we're seeing within that particular cell type. So as you can see from this plot, we see that the older programming stays different compared to the younger one and higher over the course of time. Our next question was asking how this reprogramming accumulates across age. And in order to do this, we utilized our 234 immune aging cohort and also pulled in 2 external data sets that accumulate to more than 1,000 individuals. And we quantified this RNA age metric in each individual and then plotted them over the course of age. And as you can see here, we actually find a common accumulation of transcriptional reprogramming across healthy aging. So it's important to note that all of these studies up to date have been done in peripheral blood of individuals. But we wanted to know if this aging metric actually translates into cells within tissue. So in order to do this, we utilized a publicly available data set that has lymph nodes from adult donors. And we actually looked at this transcriptional reprogramming within T cells in lymph nodes. And as you can appreciate here, we also see reprogramming within these tissues. Thus, we can conclude that this reprogramming is actually a conserved feature of aging. Advanced aging is classically associated with the development of inflammation, or often termed inflammaging. And so our next question was, do we see this reprogramming happening simultaneously with the development of inflammation in these healthy individuals? And just to summarize the data here, what we find is this is not the case. And actually in these cohorts who are prior to this really advanced stage, we do not find elevated amounts of inflammatory cytokines within circulation, really indicating that in this context, inflammation is not a driver of T-cell reprogramming. Another question we had is if you have a chronic viral infection, in this case, cytomegalovirus, if this actually impacts T-cell reprogramming with age. We know this is a very common viral infection that accumulates as we get older, so more and more people develop this infection. And so what we did is we first characterized the cell types that are actually expanded if you have CMV infection. And what we can see is compositionally, you have changes in your immune system based on this viral infection, and the composition is actually different between young and older adults. But does this impact the transcriptional reprogramming in the earlier subsets we found, such as the naive T cell and central memory compartments? And when we look at transcriptional reprogramming in these earlier differentiated populations, we actually see that CMV doesn't have an effect on T cell RNA age metric. This indicates that while CMV infection does revamp the landscape of T cells compositionally, it's not related to age-related T-cell reprogramming. To summarize this briefly, what we find is that T-cells are reprogramming across age, and this happens prior to classical older age. In particular, this also is independent from the development of inflammation and chronic viral infection. We next wanted to understand the implications of this reprogramming. Might this reprogramming link to the differences we see in vaccine response as we age? And how does it actually link to immune functionality? This is where the team began to dive into flu vaccination. And so what we did is we actually selected a subset of individuals, both young and older adults, that received the flu vaccine starting in the 2020-2021 seasons. And then their second year was in the 2021-22 seasons. This is really important because each flu vaccine actually has a slightly different composition. And so in order to normalize for this difference in composition, we need to make sure individuals received the same flu vaccine. From this cohort, we then looked at their flu-specific antibody levels. We also looked at the functionality of these antibodies using an HAI assay, as well as the different types of IgG isotypes that are elicited from these vaccinations. This is just a beautiful rainbow plot that highlights a bunch of these different parameters, in particular looking here at antibody functional responses. And what you can see just broadly is that there is a wide range of functional responses to these different antigens within flu vaccines and depending on the flu season. So how do we dive into this? We begin by looking at the ability of donors to elicit a high response to the vaccine, broken down by each of the strains in that vaccination. And so over on the left, I'm highlighting our most interesting finding, which is actually to that B. Phuket strain, which I mentioned has been in the vaccine for more than a decade. We actually see that older adults have fewer high responders than young adults. Now, this is not the same for every strain. For instance, in the B. Victoria strain, which was new to this vaccination series, we don't find differences with age. We wanted to follow up on this B. Phuket finding next. So what we did is we actually looked at the amount of IgG specific to this B. Phuket between old and young adults. And what we see is that the level of IgG they make in response to this is actually lower in the older adult cohort. We further looked into the functionality of these IgGs. So what type of IgGs are there? There's 4 different types in humans, but in particular, IgG 1 and 3 are the best for protecting against influenza infection. And so what we find in older adults is they actually skew away from this response more towards an IgG2 response, which is not as effective against the influenza virus. So those data highlighted that we actually have an early reduction in influenza responses prior to advanced age. This aligns with what we saw with T-cell reprogramming. So we wanted to understand if these two factors were related. And indeed, we find that T-cell reprogramming, specifically within the central memory compartment, highly associates with different parameters of influenza vaccine response, in particular with the changes in the Fouquet IgG ratios, functionality, and total levels of antibody. So yes, this T-cell reprogramming does associate with vaccine responses and changes in these vaccine responses across age. So how does this reprogramming actually change the functionality of T-cells? That was our next question. Memory T-cells come in many different flavors, as we like to think ice cream flavors. But actually what's really important about this is these types of responses from T-cells can alter B-cell responses, which is what we see to the influenza vaccine. And so this was our first question we had. Does this T-cell reprogramming change the flavor of memory T-cells? And indeed, we find that when we look at the transcriptional programming of these cells by age, we see a Th2 skewing in the memory compartment of older adults. We wanted to further examine this, and this is where actually this trimodal assay that was developed here at the Allen Institute was utilized. In this assay, we actually can simultaneously profile protein, RNA, and the epigenetic landscape of single cells. And so in a previously published study, we focused on differences in T cells across age. And so we wanted to utilize this data set in order to ask, do we see epigenetic landscape changes in the same compartment that matches a Th2-like skewing? So before I get there, I just want to highlight how we've used this type of trimodal data before, and in particular, why actually having RNA, protein, and chromatin landscape linked at a single cell level is so insightful. And really what we did in this previous study I was describing, we were able to use all three pieces of information collectively to actually identify a novel subset, here it's called MNP2, in children that actually goes away in older adults. And one of the key pieces we were able to use from this trimodal data set was not only were we able to define this population using protein and RNA markers, we were then able to show at an epigenetic level these cells are very distinct from all of the other T cells within this compartment. Now going back to our current work, and when we think about the epigenetic landscape, it's really important to note that each of these flavors of T cells, including Th1, Th2, Th17, have distinct transcription factors that define these profiles. And this is where we can actually robustly utilize the T-seq data to answer our questions about Th2 programming. Do we see evidence for higher GATA3 activity in T cells from older adults? Using the RNA information within the T-seq data set, we were able to label a number of different T cell types that actually matched our Immune Health Atlas, as well as our Sound Life data sets. And then we can use this to actually dive into the epigenetic landscape of these cells. And in particular, we were looking for evidence of Th2-like transcription factor regulation in these cells. And what we actually found is that in older adults, which again are in gold here, there's more chromatin accessibility with motifs for many of the Th2 transcription factors. This indicates that this is a global Th2-like skewing happening across the T cell compartment. And we find evidence of this both in the CD4 and the CD8 T cell compartments, indicating it is a global phenomenon that's happening across age. We next wanted to understand how this epigenetic change might actually lead to downstream functional changes of these cells. And so we know that Th2 cells classically secrete specific cytokines. Here we're going to highlight IL-4 as their major cytokine producer. And so what we did is we actually took T cells from young and older adults and looked both at their IL-4 production as well as the amount of cells that actually have IL-4 at resting. And just to summarize these findings, what we indeed find is that this RNA and epigenetic reprogramming of cells in older adults leads to higher IL-4 secretion, as well as a higher amount of IL-4 positive cells in older adults. And finally, what's most interesting is we find that the IL-4 secretion nicely correlates with our B-Fouquet IgG isotype ratios, really indicating that there's a link between T-cell reprogramming, vaccine response, and Th2 skewing. So just to summarize what I've shown you, our study found that indeed immune aging is not a steady decline like one might think, but a dynamic reprogramming of T cells, in which memory T cells are qualitatively different in young and older adults. In particular, we find this Th2 skewing. This alteration is happening prior to advanced age and is not driven by inflammation or chronic viral infection. We find that this T-cell reprogramming impacts functional responses to highly boosted influenza vaccine antigens. In particular here, I was highlighting the B-Fouquet antigen. And to also note, we find that this reprogramming occurs within tissue and in young adults at risk for developing autoimmune disease, potentially linking T-cell reprogramming with age-related disease development. And so really, just to summarize everything we talked about, we're really trying to understand if we can utilize this information now about T-cell reprogramming to actually improve immune function with age. And so the concept would be, from these data, might we be able to specifically target T-cell reprogramming in advanced stage or prior to advanced stage to, in the end, improve immune function and elicit better protection, knowing this information about the immune system. And this is where we're really hoping to head with this research in the future. And with that, I'd just like to thank the entire team. Here at Allen Institute, we do big science and team science. And so many people were involved in this study. And here I just circled everybody who actually contributed in some way to the publication that I just described. I'd also like to thank all the study participants for their time and commitment, in particular because these studies occurred during the COVID pandemic, and all of our funding resources and collaborators. And in the last few minutes, I really just want to highlight something in particular about the Allen Institute and the fact that we don't want to just publish papers, but we really want to promote open and accessible science. And so from this study, which you can go and find, it is previously published in Nature in October 2025. What we've actually built on top of this data set are a number of resources for the scientific community. This includes open and accessible data and information on both the Atlas and our Sound Life cohorts. And this actually allows you to go in and look at the clinical metadata. You can access the single RNA sequencing data files and read more in detail about these studies. The team also has developed a number of interactive tools to visualize and explore this data, and I would really encourage everybody to go check them out. This is a really nice way to share data in an accessible way. You don't have to know how to code. You can just go click around and actually explore the data both in the terms of the Atlas, looking at different cell types and their gene expression, as well as go into differential genes based on age, influenza vaccine, CMV infection, or sex. So please check these tools out. If you scan this QR code, it will take you to these resources, and I hope you enjoy exploration of them. And with that, I'd like to stop and take any questions that you may have. Thank you for your time.
Thank you very much, Claire. I'm sure everyone here will agree that was a fantastic and highly informative presentation, and we really appreciate you sharing your expertise with us today. So we're now going to move into the Q&A session. So to submit a question to our speaker, please enter your question into the box to the side of the live stream player and click send. We'll answer as many questions as possible during the time available. So I can see we already have some questions submitted, so let's get started. So what was the biggest surprise from your findings?
Yeah, so I think what we were most surprised by was actually how stable the transcriptional reprogramming was in the naive compartment. And really, I think this has been not very well appreciated because there's not that many longitudinal studies doing deep multiomic profiling in people. And so being able to really utilize these omic data sets to go deep, not into just the frequency of cell subsets, but actually how their RNA programming looks like over time and across age was really critical for this discovery.
Wonderful. And why are T cells the most affected immune cell with age? So what's actually, and then the second part of that would be what causes this reprogramming to occur?
Yeah, so while we don't know the direct mechanism that's causing the reprogramming, that's actually one of our active areas of research, we do know that the organ that produces T cells actually goes away as you get older. This is called the thymus. And so you have thymic involution that happens over time. And so actually what happens, instead of being able to produce new T cells, you have to maintain this compartment long term. And so really what happens is that your cells have to adapt to now being able to live for decades as you get older versus being able to reproduce this compartment as when you're younger.
And so what's actually causing the reprogramming specifically with the T cells, do you think?
Yeah, so one of the hypotheses we have is that because these cells now have to maintain themselves longer, they have to, stay alive much, much longer than before. They actually have to adapt to an aging environment. And so for us in particular, we're now actually looking in tissues using a lot of these multiomic tools to see if we actually find evidence of cellular interactions within tissues that might be driving the T cells to change their programming.
Fantastic. And would you expect any effect of the gut microbiota in either health or disease on T cell reprogramming?
So I think that is an amazing question that I would love to look more at, because we know the microbiome has a huge impact on the immune system. And I'm particularly interested in if they could be driving this naive T cell specific reprogramming. I think that's a fascinating hypothesis and one we should definitely look more into. And I think starting to look more into the tissues will help answer that question and provide some evidence if it's linked to the microbiome.
Excellent. And can we use the RNA age metric as a biomarker of immune health?
Yeah, so I think we need to do this study in more people. I think one of the caveats to our studies is that it was still done on people within the Seattle area. And so what I'd love to do is actually using a lot of these now larger, more diverse, publicly available data sets, if we could run these same type of analyses to understand how universal this RNA aging metric is, to then really be able to know if we now have a basically molecular clock that tells you your immune age, not just your biological age.
And how did the COVID pandemic affect you? Like, were you affected at all by COVID infections, maybe skewing some of the data when you were looking at your data sets?
Yeah, so that's a really good question. So because we started recruiting right in the time of the pandemic, we actually monitored infections, so we know when people got sick or not. We also know their information about their COVID vaccines, which we've just started to look at as well, and how their immune system might have been responding to either COVID infection or COVID vaccination right when the first vaccines for COVID came out.
Wonderful. And so what are your next kind of steps? What are you looking and what are you excited about in the future?
Yeah, so I mean, I get excited about science in general and using new technologies in order to answer questions about human immune responses and the human immune system. Our next steps where we're actually heading is we want to understand how vaccine responses work across the entire age range. So not just in advanced age, but also earlier during childhood, and really get an understanding of the full programming of the immune system and its functional responses across age to then be able to better tailor vaccines to work in different populations. I think one thing we're really starting to realize is that an adult immune system is not a children's immune system. And we really need to think about this more deeply as we start to think about vaccines and therapeutics and more age-related disease susceptibility.
Fantastic. So I want to thank you once again, Claire, for joining us today. So we unfortunately that's all the time we have available, but any questions we didn't get to can be answered offline as soon as possible. So the webinar will now be made available on demand, and you can access the recording using the same link you used today. We'll make sure any questions submitted on demand are passed along. A certificate of attendance will also be sent via email to everyone who attended today's session. And with that, thanks again to our speaker, Claire Gustafson, and to everyone in the audience. We'll see you again soon.